Hi, everybody. Today is Thursday, February 16th, 2023. Welcome to Neuroscientists Talk Shop, UTSA's neuroscience research podcast. We're back after a slightly longer than normal winter hiatus. We were doing faculty searches, which preempted our regular schedule of visitors and everything else that we do. And so after the lengthy and exhausting series of faculty searches, it's great to be back to our regular schedule. So today we're talking to Rakez Kayed. He is a professor in the Department of Neurology in the George and Cynthia Mitchell Center for Neurodegenerative Diseases at the University of Texas Medical Branch, Galveston, not too far from here. Rakez studies neurodegenerative diseases, the pathology underlying them, and the potential treatments that may someday make them obsolete. Hi.
Hi, Charlie.
Thanks for coming. So with us today is our own George Perry, local neurodegenerative diseases expert and longtime colleague of Rakez.
Yes.
I understand. Thanks for joining us, George.
Thank you. Really a pleasure to have you here.
Hope your hand is okay. Something bad happened?
I slipped in Coimbra, Portugal.
Wow, it happened in Portugal.
He's
the only thing exciting.
You had to come home with a bad hand the whole way home.
Fractured the finger.
Okay, I'm your host, Charlie Wilson. And Rakez, I'm hoping to learn something about tau protein today. And so tau is one of these proteins that always comes up when we're talking about Alzheimer's disease. And it is the primary component of neurofibrillary tangles, as I understand. And it's also present in lots of other neurodegenerative diseases. It's sort of ubiquitous when the brain is in trouble, I guess. And you can correct me on that if you want to. And it's present in lots of different places, but not always in the same places, I guess. And I know that we're going to talk about how tau is bad for the brain, but I'd like to start with just what tau is and what's its normal function for the healthy brain. Why does the brain even have tau in the first place?
Oh, okay. That's pretty... I wish I have a good answer for you, what's tau, because even biologists, cell biologists, are still puzzled what the biological function of tau. The only biological function we always go back to is it supports the microtubule in the cells. So wherever there is microtubule, assemble and disassembled, and these are like the highways for cells to transport things. Tau is one of the proteins supporting the microtubule. And the whole idea is that in the neurons, you need these things to assemble and disassemble fast. So there is high concentration of tau protein. And the original connection to Alzheimer's disease is that when tau is associated with microtubule and it gets hyperphosphorylated, then it dissociates from the microtubule and it kind of forms these aggregates, which we see in Alzheimer's disease and supranuclear palsy and Pick's disease and others. Almost 22 diseases you can find tau aggregates.
So if we were to knock out tau, what would happen?
That's another great question. You put me on the spot. So no, it's cool because it's one of the family of proteins. So I think many labs, we have the knockout mouse for tau, and it's viable, so it lives. However, there are recent reports showing at later age in these mice, you can see some motor deficit or some deficit, but you can knock tau down. Yeah. So
are there compensatory changes?
Not to my knowledge.
No change in MAP2 or...
I think there must be, but I don't think it's well investigated, like which compensatory mechanisms. But all biological systems are redundant. So the key is the knockout mice are viable and you can study them and you can look at them. And tau is in many cells, not in the brain. I think there is in the heart, there is big tau, they call it. So it's another
Big tau?
Yeah, it's called big tau. Check it out, but it's a big tau. So
Different
and
alternative
tau comes
splicing.
alternative splicing. That's one thing also. This is when I said the biology of tau is still a mystery, because at least it's the alternative splicing of tau releases different isoforms. And one hypothesis was, for example, in Alzheimer, you see two of the isoforms aggregating, the 3R tau and the longer, a little bit longer 4R tau. And one hypothesis was, if you can have more 3R versus 4R, you may help with the disease, but all these are unknown. So I would rather have no tau aggregates at all. But yeah, but the biology of tau is still a mystery.
So in its normal function, in which it's interacting with the microtubules, it's monomers that do that? Or?
Yes, it's the monomeric tau. At least to the best of my knowledge, it's monomeric tau. So I don't know if there is dimers. If they are functional dimers, they may be due to the disulfide bonds, because it has two cysteines, but to my knowledge, it's monomeric tau, which is functional.
So the fundamental thing that goes wrong is the formation of oligomers. Is that correct? Or are oligomers also normal and normally there?
No. Okay, so that's another trick question. Okay, so he's going deep in it. So if you are talking to an amyloid person, oligomer is an amyloid aggregate, which is not like the oligomers when a receptor assemble in oligomeric units.
So you don't think this is part of biology?
So it's not, at least to my knowledge, no. For example, there are some evidence saying synuclein tetramer is a functional tetramer, but these are functional oligomers, or multiple units coming together is different than the amyloid oligomers. Amyloid oligomers, mainly, these are the at least toxic aggregates of a protein when you have mainly beta sheets and still flexible regions, and they clump together. So oligomers, in our terms, they are clumps of proteins,
not
multiple protein units coming together.
But in the tau oligomers, those
are the toxic ones.
Those are toxic ones.
Yeah.
And how many,
At least in our hands, the ones we focus on, okay, let me just, so people know. So for the protein, there is the monomer. And assume there is the fibrillar or neurofibrillary tangles. In reality, you can put millions of monomers, units, in the fibrils. Fibrils are highly ordered structures. It's almost like spider silk. It's an amyloid, like it's a fibril. So with the fibrils, you can back so many protein units. The oligomers, which we study and we focus on as the toxic scramble aggregate, mainly 2, which is the smallest, 2 to 3 at the lower end, and 8 to 12 at the upper end. Now, somebody may argue, but you don't have 16, not 12. Yeah, we can have 16. You can find 16, 24, but their toxicity becomes less. So basically, for a protein amyloid aggregate, in order to be toxic or biologically active in a toxic way, you have to have the size right. The surface hydrophobicity now is being implicated. So there's more than one. So size of aggregate matters, hydrophobicity of the surface matter, stability of it matters. So all these factors together. But if you have an oligomer which is really large, it becomes less toxic.
What is the difference between toxicity and physiological activity?
toxicity and physiological activity. Of the aggregates, I don't think the aggregates are, I think the aggregates are non-physiological. You know what I mean? It's just like, I don't think tau forms amyloid oligomers for physiological.
So all
Relevant.
these proteins form aggregates that are all detrimental.
I really don't want to venture into the functional amyloid field because it gets tricky, because there are functional amyloid, but these are like in bacteria. Bacteria can attack other
You
organisms.
mean like curly fibers?
Yeah, curly fibers. You can do these, and curly oligomers and stuff. But I think
We're talking about on the surface of bacteria.
bacteria, there's
amyloid.
and it's controlled. So the bacteria use the oligomers which we study, or the aggregates, the fibers which we study in Alzheimer. Bacteria can use them as a biological mechanism to attack other and make, for example, make pores in their membranes, make them leak, etc. So this is what he refers to. But
Well, I'm asking the question more, if you have several proteins which have the capacity to be toxic, wouldn't there be tremendous evolutionary pressure not to have that structure?
yeah, but that's been looked at, especially at the edge of the beta sheet formation. So yeah, that's been looked at. But I think people don't
What is the answer?
know, like, let me just finish this thought because I think it's important. Like, you can count the proteins associated with diseases, tau, A beta, alpha-synuclein. But if you pick a protein of your choice, any protein you came across, and you give it to me, under non-physiological conditions, I will convert it to you to an amyloid, and it will be toxic. So the idea that an amyloid can be formed under non-physiological conditions. Now, in humans, it forms, especially with aging, they form. Doesn't mean they did not form before and the cell cleared them. But if you give me any protein, not A beta, not tau, of your choosing, I can convert it to amyloid. And this work was pioneered by Chris Dobson, the late Chris Dobson at Cambridge and others. The other thing also people don't realize, that before we used to say, okay, you need at least 2-3 amino acids to form amyloid. The work of Ehud Gazit at University of Tel Aviv now showed like even you can form amyloid, toxic amyloid, from metabolites. So basically, the idea of amyloid is a process.
You mean non-amino acids?
Non-amino acids, yeah. Yeah. But first they settle on the dipeptides, they can form amyloid structure, but now it's extended to the metabolites. And we saw it too. So
But we've just been saying that tau is toxic when it's not an amyloid, when it's not forming the fibers, but when it's just...
when it's forming the oligomers, yeah.
So really, from a tau perspective, maybe the amyloids are not the culprit, are not really the important thing to know about. The important thing to know about is why the oligomer kills things.
I think that is one thing we are excited about. So the oligomer toxicity have, okay, oligomers may have similar mechanisms of toxicity. So if you take A beta oligomers, if you take synuclein oligomers, if you take tau oligomers, they may have similar mechanism of toxicity. And we looked at them before, others are looking at them. One of the hypotheses was the oligomers insert in the membrane, and then you can have ion dyshomeostasis, or even sometimes if the membrane may be with the cholesterol, high cholesterol or low cholesterol, different membrane composition, these pores can even lead to protein dyshomeostasis. So yes, so there is a hypothesis. I worked on it, others worked on it, like maybe the different oligomers have similar mechanism of toxicity. So that is correct.
So sometimes people talk about tau as a misfolded protein. Is tau a misfolded protein?
No, that's like just, I mean, it is originally doesn't have a structure. And I think tau doesn't have a structure not because it has so many functions, so it needs to be flexible. So it's one of these like flexible proteins. And when you say
tau can
so basically in reality, when you say misfolded tau, actually you try to fold it. You start to induce more beta sheet elements in the tau when it forms the oligomers and the fibers. That's a technical term people use. And also people, I think some people don't know that in order for a protein to form an amyloid, you don't have to go all out and disassemble the native functional structure. You can change just few amino acids to form the initial beta sheet and hydrogen bonding, and the rest of the protein.
That's what I was thinking. In terms of evolution, there'd be a lot of pressure to not have those amino acids.
Yes,
but that isn't true.
That's not true, but I cannot.
I mean, if you look at amyloid beta, it occurs in higher animals.
Yes, it does.
Not in lower animals.
Yeah.
So,
like dogs have amyloid deposits.
So I'm still thinking about tau, though. I'm still thinking tau is not a misfolded protein. It's just an oligomer. So it could the oligomer could dissociate, go back to work doing whatever.
No. I think we are not on the same wavelength. The amyloid oligomers are harder to dissociate. So
why are they hard?
Because
they've had a conformational change. They're not beta. You said they're not beta pleated, right?
There is parts of beta pleated sheet,
but not to the same extent as the perihelical filaments.
No, not to the same extent, but you must have a core that is stabilizing these oligomers. And then you have the flexibility of the other part of the protein.
But those are all just weak interactions, correct?
But weak interactions are biologically relevant,
but they should be reversible, like Dr. Wilson is suggesting.
I was just...
No, I like that idea.
No, I don't think. I think we are not. I think we are not on the same wavelength. Between the 2 tau units or the 3 tau units which form the oligomer, there is some hydrogen bonding in one stretch, and most likely it's in the C-terminal or the microtubule binding domain. But we cannot speculate till we get the cryo-EM structure.
These things are stable.
Yeah,
what you're saying is
they're stable.
They're not
they're stable
reforming
as together, but the coat or the outside is still flexible. And I think this is why it can interact with many receptors, for example. It can interact with many proteins. And this sometimes will say, okay, if the oligomer adapt this surface conformation, then it may bind to an insulin receptor or heparin sulfates, or it can do a lot of things. So there is some element of surprise when you work with oligomers, that they may bind different receptors, and this is due to the flexibility of their surface. Some of them like bind to toll receptor, RAGE, etc.
Is the that surface is transformed? There's some people who think that the N-terminal region is stripped off when you actually have more C-terminal in the final PHF.
Yeah, I really don't work personally on truncated or just the amyloid forming part of tau, because I think you're missing a lot. I mean, it's informative.
Some people think it's physiological, though.
Yeah, I don't know about physiological. But
I was just thinking about also the same kind of thing about the transition from good tau to bad tau. Does it actually mean the molecule has been altered? Or is it a reversible interaction? That was my original.
The molecule is altered I don't think it's reversible. It can get degraded, and there are mechanisms, the
Proteus.
protosome unit 19, I think, was implicated, especially with these toxic oligomer, autophagy. Now you can find these oligomers in exosomes. So are they being shipped for some degradation or something? We don't know. So there is degradation. But I don't think once you form the oligomer.
Does the oligomer transform into paired helical filaments directly?
So this is what I mentioned, if I remember.
Did you cover that? I don't.
I think, so with oligomers, there is on pathway and off pathway. So there is on pathway to fibrils and bare helical filaments. Yeah, you can transform them. So
Do they do they have to partially disassemble in order to form the other structure?
I mean, there are two schools of thought. So some people suggested they have to disassemble to monomer to form, but this is not for tau, for other amyloids. But I think it's more about the conformational changes to adapt to the fibrillar structure.
And then they add monomers.
Yeah. So for example, even in fibrils, most of the fibrils which people see the pictures, they are these almost dehydrated, unique shape. But in reality, fibrils also have a fuzzy coat, and if you hydrate them, you can see them fuzzy coat. And most likely...
And those are not seen by cryo-EM, right?
Yes. But you can see that sometimes, that there is a fuzzy coat.
By regular EM.
Yeah, by regular EM. Yeah. So if I want to guess which oligomers may be worst, or loosely using the term disease relevant, I would say most likely it's the off pathway, till the cell ability to convert them into fibrils is impaired. Because I think tau aggregates at very early stages, maybe even in teenagers, tau aggregates.
So the idea is that the fibrils are kind of a sink that can suck up all of that stuff.
To one stage, yeah. Up to one stage, yeah. I think the first step, because we are the cell producing so much tau, and tau is degraded, so the first step is degrade. But if it forms misfolded monomer or oligomer which cannot get degraded, the easiest way is to back into fibrils. And as I mentioned, a fibril can hold tens of millions of units. So that's the easiest way. So it's a protective mechanism at the beginning, but after a while, it may cause issues.
So it sounds like the problem is the cell is just making too much tau. Could we just downregulate tau?
I mean...
We don't know that much about synthetic rates of tau, do we? I mean, I know people did that, but I think was it ever clear?
So when they did radiolabel for A beta, I think it was easier to detect or to decide the turnover of A beta, but it turned out tau has a longer time life than A beta, and this is why when... I don't remember.
Which studies are you talking about with A beta?
I think it was in Washington University.
Oh, those with the CSF?
Yeah. No, even blood. Even blood.
Yeah, well, I don't think that's very reliable.
But still, there is another factor.
There was the whole brain level.
But there is another factor, is like how long these things can survive.
No one did in situ hybridization or the more primitive type things where you just look at expression level.
I don't know. I think you're leading me into territories which are not relevant. Okay. I think because the relevant thing, for example, to see if neuronal activity means you have to produce more tau, because that's key. For example, if a neuronal activity requires producing more tau, then when it's, at least this is relevant to the disease.
And you know that microtubule number goes down during aging and during Alzheimer's states. You don't believe that?
I don't. 100%.
I did that.
I don't believe that. I think it's...
But other people found the same result.
Yeah, but I think the whole idea is that we go back to tau production. So if the neuron is hyperactive, do we need to produce more tau? Most likely, yes. And then after that, what happens? So the flux of the production versus degradation is key for tau.
Or could it be that you have an imbalance in it compared to microtubules? In Alzheimer's disease, microtubules are like 80 to 90% less.
I don't know.
You don't know?
I don't know. I cannot say anything.
So if that's true, though, that means tau's got nowhere to go. It ought to be interacting with microtubules that are not there, and that would make an excess of
tau, but it should be degraded in a healthy cell.
Should be.
So that's the whole idea. Tau is easy to degrade.
And that's been shown by other people's studies that tau maximum.
I understand. So you don't really buy that as that makes perfect sense.
Yeah.
Before we get done, I'd really like to bring up this issue of propagation, because everything we're talking about sounds like tau is a cell phenomenon that really doesn't have that much to do with the network. And a cell somehow dysregulates tau and starts making a lot of tau oligomers, and so that's bad for that cell. And so now, but it's often said that tau can somehow propagate down a cell and induce tau in the next cell down or in other places nearby. So what does that mean for those oligomers to induce oligomer formation in some other cell?
Yeah, so I think, forget the human prion diseases. Even if you look at yeast prions, so it's known that if you have a seed, you can do it can induce conformational changes in the other protein, and it propagates. So the propagation, so there are mechanisms. But the whole idea is that in fibers, most likely it's a templating.
But this is true for this templating. It's true for every other polymer that's in a cell. It's true for cytoskeleton proteins.
Sure, but how about oligomers? Because
oligomers they induce conformational changes.
And cytoskeleton,
and this was studies a long time, the mechanism, how it induces the conformational changes. It was studies.
So if you just had monomeric tau sitting around and you dropped one of these oligomers in, it would start forming.
Yes, every molecule will adapt the new conformation, and it will become oligomers, and actually homogeneous oligomers.
But that also is true for microtubule.
All you need for propagation then is for the stuff to be transported down the axon and released.
Yeah, and this is, I mean, there is the distal spreading versus the local spreading. Distal spreading is what you described. So basically, if a seed is transported via the axon to another region of the brain, then you start propagating the other region. And this is the distal spreading.
It's the pathology, the progression to the brain fits as if neurons connect.
But what about?
I don't know about being reliable.
But what about brain injury? That will start as localized spreading, and then it will spread from that local area. What if a neuron died and there's some aggregates there, or a cell died? So we cannot just be transfixed on the axonal or distal spreading, because there's so much to know. Because if you have tau oligomers in one brain region, which is connected to three brain regions, are all these three brain regions going to get tau pathology? Is it regional dependent, the propagation? Is it cellular dependent? We don't know. So yes, as a basic mechanism, the seed will go there, and in theory, it should corrupt that tau in that area or in these cells in that area. But there is regional and cellular specificity.
The church institutions of northern Iberia at the time did have contact.
We have another podcast
contact with the Church of Rome, and so whenever there was reforms, like when it was being pushed by the Franks,
I was listening to a podcast myself this morning.
Those were kind of just being drip fed into northern Spain, while Andalus.
I'm sorry.
No, it's okay. You are international.
It's a podcast on podcasts.
Okay, Professor Barry is an international ambassador for Alzheimer in the whole world. So that's thank
sorry.
you. No, it's fine.
I don't want us to quit without talking something about what are the prospects for therapy of tauopathy. So I know that there are lots of Alzheimer's disease has lots of things. You can think of different ways to attack it. But if we're just thinking about from a tau perspective, how do we cure tauopathy?
I think, personally, I would go either immunotherapy or polymorph or strain-specific immunotherapy or aptamers, some molecular approaches. That's one thing. Having said that, you know, there is antisense oligonucleotide is being tested. This goes back to your idea. Just lower the damn tau completely and see if you prevent it. But, you know, there is issues with the delivery, etc., with these antisense oligonucleotide. Now, another idea, which I think now it's not going to be valid, was to convert, for example, for Alzheimer, 4R tau to 3R tau. But now we know that 3R is as bad as 4R could be, or at least it's still bad. So there is the small molecule approach. There are clinical trials that disassemble tau aggregates or prevent. I think...
For disassembling, how does that work?
I mean, there are some data showing that there are small molecules that disassemble the fibrils.
Oh, I see. So you could just...
Fibril is a polymer.
Try a zillion different molecules and find one that causes the oligomer to fall apart in vitro.
Yeah, but the problem, if you disassemble the fibrils, are you going to get
even
more or toxic, or you get inflammation? So I think the best approach to think about is to try to find the aggregate which forms early in the disease and target it as early as possible. You know what I mean? It's just like, don't let it till the disease progress and you may have different structures, many aggregates, and then, but if we can find the early aggregate and we can specifically target that, that would be the ideal approach.
What would be a way to target that? Is that an antibody?
I think antibodies are being used, you know.
You said that some of them had benefit? I'm not aware of that.
The tau one, no, not to my knowledge. But at least they're promising.
Promising. People are not dying.
Promising is not stage 3. Promising is stage 1 and 2. I mean,
you mean they didn't die?
Yeah, they did not die. But also there is no...
That's a pretty low barrier.
But why? Why? I think you have...
I mean, because the amyloid kills people. So
but we have to be optimistic. Okay. When tau immunotherapy started, 2007, the first preclinical testing compared to 1999 for A-beta. So the tau field is catching up. So I'm optimistic that there will be.
But if we were to enumerate the possibilities, one would be to downregulate tau by attacking it at like with antisense RNA or something like that. And another one would be to try to dissociate the oligomers.
Yeah.
And maybe they would...
Or convert the oligomers into larger non-toxic aggregates. That's,
What if the amyloid's a response to the problem and you're disrupting the response?
You mean the amyloid formation is a response to a problem?
Like inflammation.
Yeah, I think, but still, most of the inflammation in the brain is associated with protein aggregates. So you
only?
But there is some. Astrocytes have amyloid and have...
astrocytes actually protect the amyloid.
I mean, protecting in which way?
They lay chondroitin sulfate on it so the microglia can't remove it.
But that goes to the early stages, and after that...
What do you mean the early stages?
Like, if you think that plaques and tangles...
Whenever there's amyloid deposits, there's chondroitin sulfate laid by astrocytes.
Yeah, because they are activated by amyloid.
Mm-hmm. Better than anything else. And when they get activated, chondroitin tells the microglia they can't degrade it.
So, George, are you saying that that's a therapeutic approach somehow to ramp that up?
No, I'm just on this part of the biology that happens, and it actually is making the plaque into scar tissue.
I think,
It's being used as
but
a
the
therapy
question was about therapeutic.
Mhm.
I think it's still unclear. Upregulating the immune response or downregulating it is beneficial.
Or the removing amyloid of tau or amyloid will be beneficial.
Yeah, I don't
So
sign
easy,
for this, but that's George's
of course, if we
opinion.
can't do something, we can't tell whether it's good or not, right? So
You have to do the
the
test.
first thing you have to do is figure out how to get rid of tau, and then you find out whether getting rid of tau is helpful or not.
Correct.
So I was just asking about how to get rid of.
Yeah, that's what I answered. It's helpful, but you can do antibodies, aptamers, small molecules. You can activate the proteasome, autophagy degradation. There's a lot of methods.
A lot of these things
There's a lot of approaches. Yeah.
Great. So we can look forward to a day when we don't have any more tau than we want, and then we'll find out whether.
Exactly.
I agree. And one other thing, I think the tau is being used as a biomarker for the efficacy of some clinical trials or in the blood, actually. That's also exciting now. So people measuring tau 217, tau 181, and also there is even evidence from the clinical trial that this can be to evaluate if the trials are beneficial, or there is, at least if there is target engagement. Let's not overreact.
Okay, well, thanks very much. I think that's a happy note to end on. Thanks, Rakez. And George, thank you, George.
Thank you, Charlie, for organizing
Thank you, and thank you. It was a nice visit, and I enjoyed all the discussions.